Pleasure to invite Dr. Rajesh Dixit for the next talk. Dr. Dikshit is a professor and focused on epidemiology of cancer for several decades now, actually. And his expertise lies in cancer epidemiology, molecular epidemiology, surveillance, cancer registries, and is a director of Center for Cancer Epidemiology at ACTREC, Tata Memorial Centre, Mumbai. Dr. Dikshit, looking forward to hearing from you about Indian Study for Healthy Aging, or ISHAs.
Okay, thank you very much for the kind introduction. So I'm going to talk about the Indian Study for Healthy Aging, which is conceptualized by the Center for Cancer Epidemiology around 2014, and now it has almost 7 to 8 years of working since then. So the study actually, there are only two study locations right now. It is in the Barshi, which is in the district of Solapur in the Maharashtra states, and it covers 362 villages and three small cities, including the Barshi town. And then recently, we have also started since last 2 years the study in the Varanasi. And those who don't know where is the Barshi, here is a map where the study location. It is in the Maharashtra, southern Maharashtra, very close to the Karnataka, and Varanasi, you already know. So the basic thing is that we also want to expand it to the other center where Tata Memorial Hospital is located. So Tata Hospital is located in Mumbai. So we have the study not in Mumbai but in Barshi, and we have started the study in the uh, Mullanpur, and we have started the study in Varanasi, and the Guwahati and Visakhapatnam is still yet to be started. So these are the two sites are actually functional. The Barshi and Varanasi and Guwahati are functional. So why we want to study, I think it is already covered by earlier talk, but just want to stress it again that we know the burden of disease very well, but the burden of the lifestyle, environmental and genetic factors as a risk factors and as a cause factors is not known, is not properly studied. And because the various parts of the India has a different exposure, it is possible to capture some of the exposures which are not very common in some parts in globally or in some other parts of the India, because there is a disease heterogeneity. So it means that there is a differences in the exposure prevalence which is causing the disease. So just again to repeat that the burden of disease is different in India, cancer, stroke, cardiovascular diseases, and there is a heterogeneity in the disease burden, but these reasons of the differences are not known. And we can mainly say that the differences are because of the prevalences of the exposures, lifestyle, environment, and genetics. So we need to find out what are these differences in the prevalences and what are the new exposures which might be present in the different community. So far globally there are a few studies with blood samples, intravenous blood samples collected. So like UK biobank, then there is Iceland, Kaduri, China's biobank, Mexican prospective studies, Cambian National DNA biobank studies, which are studying the causes and the risk factors of the diseases. So what we started the cohort studies 4 years, like now 2014, with the target recruitment is to have the 500,000 individuals from these centers. And currently 200,000 individuals in the age group of 30 to 69 are enrolled from the Barshi. And detailed lifestyle information, very similar to the LASI or the UK Biobank, which includes the past medical histories, the blood pressure, the diabetes, the tobacco chewing, smoking, alcohol, mental health, everything is collected. And this is collected on almost 70,000 individuals from Barshi and 15,000 individuals from Varanasi. And with the blood samples are also collected, intravenous blood, and they are stored in the biobank. Then we have also started the sub-studies, which is much more focus on the obesity and the DEXA. So the sub-studies, which has a measurement of the DEXA, and then mental health, there is a SKEET clinical questionnaire is being used, and the physical activities is being measured by the wearable devices, so that we can have the more proper measurement of the exposures. And repeat collection of the blood samples and urine collection is sub-studies. So out of 70,000, we plan to have almost 20,000 repeat collection of the blood sample and urine collection. And so far 10,000 repeat blood and urine samples are available. And then, of course, we also want to measure the obesity at four different points of the time, so that how the obesity patterns is changing over the time can we find it out. So what are the key features of this cohort? It's a laptop-based questionnaire, so the study is on the laptop. We have established infrastructure, which is the automated biobank is there, which has the capacity of storage of 3 million samples. We have the molecular epidemiology laboratories, which has the facility of doing high throughput genotyping, as well as the high throughput sequencing. We have established IT infrastructure for analyzing the large-scale genomic data. We have started academic programs, which is including MPH and PhD programs. We have hosted graduate and postgraduate students, and there is an established collaborations with Oxford University and even with the NCI. So what are the key measurement that we have done? So the blood pressure is measured 3 times, once at the time of the enumeration, then when we call them to the clinic, and the third time when we do the resurvey. So the blood pressure has been measured by the Omron machine, digital thing, 3 times, weight measured 3 times, height is measured 3 times, waist to hip ratio is measured, grip strength has been measured 1 time, blood collection has been taken 2 times, intravenous blood, urine collection 1 time, nail is collected, body fat is measured using the Tanita machine in the field. Then we also use the DEXA for body fat. We have also piloted using the stool for shotgun metagenomics. So the 500 samples of the stool has been collected, and now we are waiting for funds to start the shotgun metagenomics. And of course, we are using the variable to measure the physical activity on 20,000 individuals. So this is a sub-study. So this is the basic measurements that we are doing in the study. And so this is the design that and the results of these studies are, there are 3-4 publications, and the latest publication is in International Journal of Epidemiology in 2024. You can see the basic prevalence sources of the hypertension, obesity, and chronic diseases, everything very similar what was just described in the last study, that many of the individuals don't know the hypertension status, many of the individuals don't know their diabetes status, and of course the 10 to 15% is a self-reported, but when you measure it, it's much more. But to say that it's a hypertension, I think we're required to have 3 basic measurements as per the cardiac society guidelines. So therefore, we are not calling it as an hypertension. So this is a 2-step study that we do the enumeration, then the lifestyle questionnaire is being asked, then we call them to the clinics to do this physical measurement, and then the resurvey after 3 years, and the follow-up is done after every 3 years. Active follow-up is done, and then passive follow-up is also done. So 3 years follow-up on enrolled participant has been completed, and we have identified like 324 cardiac events, 208 stroke, 255 kidney diseases, 140 cancers. These are the chronic diseases were identified after the 3 years of the follow-up. So this is a basic layout plan that how the study is done. You can see the field structure that there are 4 PSCs in the Barshi. It is a similar thing in the Varanasi. I just give you the one example. And then we have the field team. There are actually 4 teams. There are 2 coordinators which monitors these activities. And then there is a lab technicians. And the samples are collected and transported to the CCE for the large-term storage. So they have the vehicles, they go by the vehicles, and this is the procedure for the enrollment. There is a enumeration, there is a consent, then height, weight, then we invite them to attend the clinic, and then the clinic is set up, and the 5% is resampling. So on 5% individualing, we again re-interview some of the individuals on some of the questions. So that question is also randomly selected, and individuals are also randomly selected. Then this is the cohort study layout in the clinic. So in the clinic, what we do is we have the Tanita machine. Now we are also doing DEXA. We have the peak flow and the grip strength there. We have stopped the peak flow now after COVID because of the variety of problems. So it was done up to 2021, but for last year, we are not doing it. Then the intravenous blood collection is done, and the health report card is given to them. So this is the clinic thing. And this is the interview, this is the house-to-house interviews. I think it's very, again, similar to the LASI protocol. They go with the laptop, and the data comes immediately to TMC server, and all these informations are asked for to them. And total time for the interviews is almost 25 minutes. And we have the specially designed FFQ after 24 recalls and everything. We have designed FFQ, which is very peculiar to the Barshi. So same thing is being done for the Varanasi, but in Varanasi we are not inquiring on the FFQ so far because the food frequency questionnaires will differ between the places. So therefore, the rest of the questionnaire will remain the same in all the centers. And then this is the laptop just to show the Omeron machine, the grip strength machine, and this is a Tanita for the body composition that we are using, spirometry that we are using, the freezer at the local in the Barshi, and then these are transported for the automated biobank. So the main features of the biobank that it can store up to 2 million samples at minus 80 degrees centigrade, and it has the automated storage system and automated retrieval system. So there is no manual intervention. You just have to keep the samples at the window, and then the robot will pick it up and put it at particular place. And when you want to retrieve the samples, you just need to give the ID number, the barcoded, which is barcoded, and then the robot will pick the samples and give it back to you. So it's a completely automated retrieval sample. There is sufficient redundant options in this biobank, and it will be expanded to store up to 3 to 4 million samples in future if the cohort further increases. So we have done so far the downstream application. The whole genome sequencing on 2,000 samples has been done, and now the quality checks and everything is in the process. So main purpose of this genome sequencing is also to develop a reference panel, because it's still very costly to do whole genome sequencing on all 50,000 or all 100,000 individuals. So the idea is to do the genotyping, which is less costly, but then because we don't have proper imputation panel, we have to use mainly the US and UK imputation panel for the imputation. So if you have the reference panel of the Barshi itself, then we can impute it more properly and accurately. Even the genotype individuals with a rare allele frequency can be imputed. So therefore, this sequencing would be very helpful in addition to other advantages of sequencing. But the main purpose we want that we will have our own reference panel, so that then we can impute it when we do the genotype on the large scale. So that is going. And then the biochemical analysis has been done, the lipid, mostly the lipid profile, the renal profile, and the HB1AC. So now in the subsamples, we are also doing the urine, microalbumin, and creatinine to detect the early kidney diseases, to see the prevalences of early kidney diseases. And then future plan is once we have the imputation, we can do at least 40,000 individuals genotyping, so that correlate it with the various diseases and obesity and hypertension and other things. And for the deaths, we do the verbal autopsy. So when in the follow-up, if you observe any death, we do the verbal autopsy. And so far, there are 4,000 deaths have been recorded. And these deaths then are linked with various biomarkers and the various lifestyle questionnaires. So for the follow-up, there are various ways we are using. We have the cancer registry in the Barshi, so which is run by Tata Hospital. So we'll link the cancer registry data with this cohort data. The Aadhaar card linkage is being tried. The hospitals, there are 5, 6 major hospitals in the Barshi, so we have access to their record. Of course, we want to digitalize them also, these hospitals, to strengthen them so that it becomes much more easier. But currently, we are just manually recording the deaths, and we actively do the follow-up, as I said. And there is Rajiv Gandhi Health Insurance Scheme, and that data is again linked with the cohort data. The death registered, we go to the municipal corporation office, and we try to link the deaths which are occurred, death certificate which are there, to with our the participants who are enrolled in the cohort. So there are various ways of the follow-up. But mainly the follow-up is done by the active follow-up every 3 years. But there are other things like cancer registries, which also gives you the passive registration and passive follow-up. So the focus is that to complete the enrollment of at least 100,000 individuals. Right now we have the detailed with the blood information on 80,000 individuals. So maybe in the next 2 years, at least 100,000 individuals should be done. Then there has to be more passive linkages that we are working on instead of the active this thing. And the other thing is how to measure the outcome. That's a major problem because we don't have the very good cause of death registration system. And so even if it is written as a stroke, we don't know the type of the stroke, and we don't know the we don't have the very good quality medical records. So that further detailing, because if you are doing a very high throughput downstream application like sequencing or other thing, you require a very nice measurement not only on the exposure but also on the outcome. And then if you do the verbal autopsy and then with a poor cause of death certification, it becomes very difficult to have the very good outcome measure. So you know the cancer of the breast, but then you don't know further details. And when you have such kinds of protein and genomics analysis, I think most important thing is to have the very good proper outcome measures is also there, not only the exposure measurement. And of course, how to further developing the data storage and other thing. And now we are doing a sub-cohort studies within that. So we have one study on the obesity there with a small number with 10,000. So it's a case cohort studies we are doing. So once you have the cohort, then you can do a multiple case cohort study. The one study we are doing obesity, other we have started air pollution study with NIH. So one can start many case cohort studies in that. That's the plan. Yeah. And so in the next 2 years, I think we will have the all-cause mortality through verbal autopsy. Risk factors prevalence is already there, and the sequencing data will also be available in the next 4 years. So these are the major challenges. I already pointed out that health outcome is the major thing, that how you measure the proper health outcome. We can have measure very good exposure, but then how outcome measurement is very important. And how good we can do the linkages. So in future, like with the help of IIPS and ICMR, if the Aadhaar card data can be used to the linkages, I think for the, at least for the health outcome, it would be great health, because we know now the Aadhaar card is used in all the cremation ground and everywhere. And in many hospitals uses the Aadhaar card numbers are there, and now Aadhaar card is also there. Most of the hospitals are also registered. So if there is a way that we can link this Aadhaar card information, the health card through the ministry support or other thing, at least for this kind of big studies, I think that would be very helpful to look after the data. And another challenge is the repeat measurement is very important, I think, because now if we are talking about the proteomics and the artificial intelligence and so many things, I think they require the repeat measurements of obesity, repeat measurement of the behavior, the mental stress, and how this changes over the period of the time. That's very important. And that's why it's not only the 1 point data, but you need a repeat measurement of the one measurement. So that's why we thought that we will measure the obesity at least at the 4 point of time. So even currently the obesity is measured at 2 points of time, but maybe if you have 4 or 5 times of the obesity is measured, or now there are many apps which can measure, individuals can measure their weight in the weighing machine, and maybe every week they can they can put up their weight, online, and these are all individual participants. So that will also give you a lot of information to work upon when you do the actual genomic and sequencing analysis. And enhancing, so more and more biochemical measurement, as you say, we just do the lipid and the renal profile, but we know that there are many more biochemistry parameters that might be of interesting depending upon which expertise you belong. For cancer, there are many biomarkers which we are interested, the others are interested in many other biomarkers. So of course, it requires funding or everything. So if it is there, you can do. The genotyping, I think that's a very important thing to do in India, because most of the precision medicine is based on the genotyping information which is coming from the Europe and US. And if you are able to do the genotyping, and now I think even with the dry blood spot, the sequencing thing can be done on the Illumina platform. So I think it will be very good to have this much of genotyping information with this good exposure and with the outcome data. So of course, the funding is another challenge. So, just to last slide to summarize that the plan, that we are in the recruitment phase, which is almost over. We have done the follow-up of almost 80,000 individuals for 3 years via various mechanisms. Now, for the genotyping and sequencing, data cleaning and this thing is on the way for at least on the subsamples. And once this we have enough data and enough numbers, something in the pocket to disseminate, then there is a plan to disseminate this data to the public health. But of course, before dissemination, we want to make it sure that it's a good quality, and we have something in our pocket before this. We don't want to disseminate empty pockets to everyone. So thank you very much. Thank you.
Thank you, Rajesh. Dr. Rajesh, thank you so much. You're actually bang on time, rather early on the time front. So we can have a bunch of questions if people have any question right there.
In case of Lasi, we could see clear divide between urban and rural. So if you take Barshi, how is that like urban rural thing? And
So, yeah, because the Lasi in terms of heterogeneity, it is more because they have the rural as well as the urban samples. The Barshi, there are 362 villages and 3 small cities. So the Barshi is a city, so you can differentiate between the Barshi and this thing, and even the Varanasi, for example. So Varanasi town is a big town, but then there are many villages around the Varanasi. So we have the small population which covers the city part, and surrounding population is the rural part. So maybe you can differentiate between the rural and urban, but they are very close because main rural is the rural which is away from the cities, many kilometers away. Because if the rural area is very close to Delhi, then it's not actually the rural. It all have the characteristics of the urban.
Yeah.
So that's important. So from I think rural point of view, the Barshi is very well covered. Barshi is city, but it's not like Mumbai or Delhi. So if someone wants a real city, then maybe someone needs a data from Bangalore or Mumbai or this thing. They are very different from these smaller town in India.
Any other question from the audience? Yes, right there.
Hi, sir. Thanks for this wonderful talk. I'm a researcher in the pre-final year of my PhD, and my question is that when we try to think of conducting these kind of experiments at a very broader scale and in a very less amount of time to get the very accurate and good amount of data in a country like India, basically, what are the biggest challenges that you think will be there, and how we can tackle with those?
So the one challenge is how you motivate the society. Whenever any society you go, the first challenge is you have to communicate with society properly, and society has to agree with you to give you the data properly and sit with you and this thing. That's one of the biggest challenge. The second challenge comes if you are collecting the blood sample, intravenous in particular, the transportation of the samples, because the samples has to be even transported within 7 or 8 hours at the 4 degrees centigrade. Otherwise, you carry the blood and the blood will not be of any use. Even if you collect the dry blood spot, they have the experience. We have some experience of dry blood spot. If it is not properly dried and if the circle is not properly filled, then even the dry blood spot, you think that you have collected, but then it is of no use. So basically, these are the problem of the communications with the society, problem of transportations of the samples, and problem of getting the skilled manpower who can work in the rural areas. This is the third problem because you get many good people, MPH and everyone in Mumbai and Bangalore maybe, but for them to go to the Barshi
Yes.
and collect the information or even to coordinate the thing, it's very difficult. It's very hard time for us. Many of the MPH and PhDs who did PhD under us, with the assurance that they will work in the Barshi, they just left after 1 year once they get some experience there. So that's, I think, getting the skilled manpower is the third challenge.
I completely agree with you. Getting skilled people is the most difficult part of study of this scale.
Yeah.
Any other question from the audience? I actually have a few questions. You're looking at correlation with cancer and all. Are you in the blood which you're collecting? Are you doing detailed hormone panel analysis? And you're like trying to understand what kind of hormonal changes occur?
Yes.
Is this something which is built into your scheme of things?
Yeah. I think yeah, that's a very important thing. We have not thought about it, but that would be very important, particularly when the breast cancer are rising.
Yeah.
And then what are the hormonal thing? Um, uh, I'm sure that for the hormonal changes, the 7 year delay in the blood collection may not affect that much. I'm not sure about that.
It all
But
depends on how you store your sample.
yeah,
Sample is stored well.
so because we have done the aliquoting, so we have made the 7 aliquots. So we collected 6 ml of the blood, and then we did 300 microliter of this thing. So the repeat thaw and this cycle has been avoided. So I think then if it is good for hormone, because
Yeah.
many of the hormone and even the RNA person say that no, we can't take like 7 hours, because it takes 7 hours from Barshi to Mumbai to transport the blood. So even if it is
Exactly.
cold chain, yeah.
So that's interesting.
Yeah.
And the second thing, which again, the question which I asked the previous speaker about incorporating the digital health using digital health platforms in this survey, like how much people use, rely, consult, or get information from digital devices as this trajectory has changed over subsequent usages.
Yeah, to change the question, you means that when you do the research,
When you do the survey, do you capture that
we will keep the we capture the information, and that time we can use yeah, how much you can rely. The question is like
No, the question is like, do you capture the information as the person whom you're collecting the data from? Are they using any type of digital health interface as a digital health
yeah,
platform?
that question is not there, whether they are using. But that's a good idea. I just got that. Yes, we need to ask that question also.
Exactly.
The availability of health infrastructure.
Yeah, and actually, where do they get the
Yes, yes.
information from?
From that, yeah. So maybe in the next time we will
True.
include that. Thank
Uh,
you. Thank you.
There's one last question here. Yeah.
Hi, this is Gaurav this side. So I have a very small technical question. Like, these numbers, 100,000, these are always been a bottleneck when we see the data from a, data analytic perspective. So since you have already covered 200,000 among these
Blood sample is only 70,000.
70,000, that is also a good number. So I was just thinking, are you also leveraging the LLM models trained on UK Biobank? At least they don't give the data, they give the models in public. And also there are many large scale models are there. Are you also thinking like leveraging this 70,000? Can you synthesize the data with the same noise, what you are going to get 500,000? Similarly, the universe is big.
Yeah.
So I'm just thinking, is there is some thought in these lines, like to generate synthetic indianized data by learning from this 70,000 data, so that, and you always have a check. Future data, you can do the correlations and keep on checking.
So yeah, so you're right. So AI is now emerging, and for last 2-3 years, there are many companies with the AI. And then I think it will be very good idea to look into the algorithm and the models which could predict also that what could be this thing, and then compare the prediction and actual data set. That could be, yeah, we can talk about it, and obviously we would like to work.
Thank you so much. Thank you very much, Dr. Rajesh, for this interesting talk.